invite Ella Fishman from the Tel Aviv University from Israel. The Fireflies of Israel, Taxonomy, Ecology, and the Potential Effects of Light Pollution. And I'm very, very curious, and she knows, because Ella has this Lampiroidea in Israel, which is also present in Greece and so on. I've been several times to Greece to look for this Lampiroidea, and I'm very curious what you found in Israel. So go.
I'm happy to tell you about it.
Yes.
Next to my bathroom. Right. Just a minute. Okay. Hello and good morning. I'm very happy to be here today. I will talk about part of my research in the master, which focused on the taxonomy of fireflies in Israel. As you know, most of the knowledge of the firefly fauna in the world is restricted to North America, Europe, and part of Asia, as can be reflected in this map, illustrating the four main causes for firefly decline in the world. It can be seen that no data has been gathered from Africa and the Middle East, due to the fact that those parts of the world have hardly been studied. In our study, we attempt to fill part of this gap by constructing the first comprehensive study for fireflies in Israel. Israel lies in the crossroads between three continents: Africa, Europe, and Asia. As a result, it has a unique fauna and flora influence from those three continents and from the diverse habitat, as can be seen in this satellite map. Many of you might be under the wrong impression that Israel is a total desert, and it's true for the southern part. But as for the center and north part of the country, we have a Mediterranean climate with some nice green aquatic habitats, as can be seen. And you can see some more photos of the Mediterranean view. This is in fact the view from my parents' windows, like a few days ago. This is Mount Carmel. And we also have long, hot, and dry summers with short, relatively wet winters. And Israel is extremely crowded, with agricultural areas in orange and settlements in black. You can see that the most developed area are also the one with the Mediterranean climate, which limit the distribution of fireflies and other animals prefer this type of climate. So it leads me to the challenge we faced attempting to study the fireflies in Israel. And the first was that the Israeli fauna has never been studied properly, and in particular, the genus Lampiroidea, which its distribution is limited mainly to the East Mediterranean. In addition, we couldn't reach to most of the types, but we did manage to get photos of some of them from the Natural History Museum in Paris, and you saw this picture before. This one is one of the recently described species collected by Pick almost 100 years ago. The description has been made based mainly on color and size, characters that are not informative for those species. And fireflies are really seasonal in Israel, and they are pretty rare, which means that many times I went into the field to look for them, and I couldn't find any, even if I've been in the right season and in a suitable habitats. So that was one of the problems. Prior to our work, 8 species in 3 genera and 2 subfamilies were known from Israel. In Lampirine, Lampiris nervosa from Lampiris, in Lampiris, and 2 species from Nyctophila. In Lucialine, only 1 genus, Lampiroidea, with 5 described species. For non-experts, males from those 2 subfamilies might look similar, but if you put them side by side, it's much easier to distinguish among them due to the significant difference in size. And the first to review the Israeli species was Olivier in 1907. He worked on the Lampirodea males and based mainly on characters in the pronotum, such as size, shape, and color of it. Those characters found to be unreliable for distinguishing among those species, and we cannot use them. After him, Guy Stard worked on the Lampiris and Nyctophila males, and he used mainly characters in the genitalia, in the male genitalia, and those found to be informative in the species level, we could use them. Prior to my work, this was the Lampirid material that was available in the standard Natural History Museum, which are national collection, insect collection. You can see it here. So it was very limited, and most of it was unsorted and unlabeled. Hence, our first aim was to clarify the taxonomic status of the Israeli species. The second was to associate females, males, as well as larvae. Data wasn't available for most of the species. The last aim was to create a key for the fireflies, the Israeli fireflies, for the first time. To achieve those aims, we conducted comprehensive collecting effort throughout the country, and add specimens to those who were already in their collections. For those specimens, we did a detailed morphological analysis, but due to the fact that most of the species cannot be distinguished using only morphological characters, we also did a molecular analysis and sequenced part of the mitochondrial genes 16S and CO1. Those found to be informative in the species level. I collected larvae and rocks during the day, and during the night I collect them from the ground together with females, thanks to the light they produce. Males I collected with nets or with light traps, that can be seen. Some of the males were trapped into spider webs, and I could collect them from there and actually steal those spiders' meal. And you can see the localities where I managed to collect fireflies during the 2 years of study. Many times I went into the field and couldn't find any fireflies. So that map presents only the localities when I collected at least 1 specimen. You can see I collected from most of the Israeli Mediterranean region. Besides of our field collection, we also published a citizen science project in collaboration with our Society for Protection in Israel. And in this project, we asked the general public to report about fireflies they spotted during the 2 years of study, and to attach the photo if possible. We got more than 2,000 reports, and after sifting them, I found out that many of them were incorrect, having reported about other insects or just piece of glass reflected in the, yeah. So we ended up with a little bit less than 1800s. You can see they are in all of the Mediterranean region in Israel. Our data shows a peak of fireflies activity during the spring and the summer. In this graph, x-axis presents the months, and the y presents the number of individuals reported or collected. In orange, males, in green, females, and in gray, larvae. You can see that in the beginning of the season, mainly males were reported, while in the end of it, mainly females. That, together with my observation in the field, proved that in Lampirodea, males are easier to detect, or to see in the field, and they are active in the beginning of the season. As for the other two genera, Lampiris and Nyctophila, females are easier to detect, and they are active in the end of the season. As for habitat, we asked the reporter to identify in which habitat they spotted the fireflies. We were not surprised to see that most of them reported it as natural. Yet, we had many reports from gardens that, together with the fact that all life stages were reported from those gardens, made us believe that those gardens can be a suitable habitat for fireflies. In the taxonomic part, I used several characters, among which the males' genitalias found to be the most informative ones, and in particular, the paramers and the diagonal shape. As for the molecular part, I used 16S and CO1 genes, and in this tree you can see every species in different color, and in orange, species that were obtained from gene bank. We use Futorinae as our group, and you can see the two subfamilies, Lamperinae and Luteolinae. In Lamperinae, we had these two genera. In Lampiris, only Lampiris nervosa was known from Israel. In our tree, we found 3 species. In Nyctophila, 2 species were reported from Israel, and we found only 1. As for Lampirodea, 5 species were known from Israel, and we found 8 of them. In addition, we associate males and females, as well as larvae, and our data indicate that all the larvae that were reported from Israel were from 1 genus, only from Nyctophila. In view of this finding, we looked for Lampirodea larvae, and we ended up with 1 larvae from 1 species. This is the first record for Lampirodea larva in Israel, and as far as we know, one of the very few in the world. In addition, 3 species were not found in this way, in our collecting, and we found 6 new species, 4 from Lampirodea and 2 in Lampiris. As for Nyctophila, 2 species were reported from Israel. We found only Nyctophila syriaca. That's found to be the most common species in Israel, and it was common in all the Mediterranean region of the country. As for Nyctophila libani, we couldn't find anyone in the field, any specimens in the field, but there were 2 specimens in the collection. 2 of them from Mount Hermon, which is the most north part of Israel. We also did an IUCN risk assessment for those species and found Nyctophila syriaca as least concern and Nyctophila libani as endangered. We also reared some of Nyctophila syriaca larvae in the lab. We fed them with snails. They grow successfully and pupated. You can recognize the males by the long wings they have. To our surprise, most of these larvae turned out to be females. But yet we had some males, few of them. And to our great surprise, those females laid eggs, females, without any mating. And from those eggs hatched a second generation of larvae. And as far as we know, it's the first record for parthenogenesis in the family. Yeah, it was very interesting to see it. As for Lampiris, only Lampiris nervosa was known from Israel. Our data shows 3 species. When we looked in a morphological difference between the males, we couldn't find any, but we did found it for the females, which were different in the size and shape of the wings. Those species were also different in their distribution This is a nice example for closely related species that can be differ only by characters in the females. As for the last genus, Lampirodea, we managed to find 4 new species, among which Lampirodea sp4 is unique by its orange pronotum and by a unique shape in the diagus. That was found to be only in the Mediterranean region. Another new species was Lampirodea sp3 that was unique by a spike in the diagus. That one found only in the north part of the country. So I want to summarize all and say we found at least 11 species of lampirids in Israel. 6 of them new to science. And we found that Lampirodea species are active in the beginning of the season, while the 2 other genera are active in the end of it. And its gardens can be a suitable habitat for fireflies, and it's important in case of natural habitat loss. We also wrote the first key for Israeli lampirids. And for now, you can use it only if you can read Hebrew, but we will translate it and publish it soon. And eventually, we also found the first record for proteogenesis in the family. We also did a lab experiment to check the influence of light pollution, but I have no time to talk about that now, but you can ask me about it later. So thank you so much for listening, and I'm glad to have some questions. If you have any, that's the time. You can come later.
Thank you so much.
Thank you.
You did it in 2 years.
Yeah.
So much work. And there are still questions. Sorry.
Thank you.
So, Ella, I have a question.
Okay. Great.
Yeah, that was really excellent work. So I have suspected that parthenogenesis happens in other species.
Yeah, I guess it must be.
And it's mostly based on observations of really large genome sizes in closely related species, because these parthenogens can't clear, like there's no selection to clear repetitive elements. And I was wondering if you had any plans to look at genome size in your group.
I couldn't hear the end of the question.
I was wondering if you had any plans to look at genome size in your group.
I sadly say that for now not. How it will be in the future, but I'm sure there must be proteogenesis in closely related species. Very important, but I asked people and I asked people sitting here and no one